Okay, I think we are going to start. It is my pleasure to introduce today's speaker, Dr. Geraldine Merle, an associate of chemical engineering at the Polytechnique here in Montreal, and a prominent researcher in the musculoskeletal health rehabilitation and medical technologies axis at the Research Center of St. Justine University Hospital. So Dr. Merle also holds the esteemed TransMedTech Research Chair in Diagnostic and Therapeutic Technologies. Professor Merle's academic and industrial career has been dedicated to multidisciplinary projects that significantly advance healthcare in orthopedics, biomaterials, and medical devices. Her innovative work spans sensor technology, smart needles, and tissue engineering, particularly in anterior cruciate ligament repair. Notably, her group was the first to demonstrate the effects of daily code application on bone growth using a murine model. Dr. Merle's investigations extend to synthetic implants and the influence of metallic ions like boron, iron, and zinc on in vivo bone formation. Her expertise in nanomaterials has led to the development of bioresorbable 2D nanostructures that enhance osteoblast adhesion, proliferation, and implant strength. In addition, her work encompasses advances in angiogenesis for bone healing, infection prevention through novel sutures, and electroceutical gels. Since her initial appointment in 2017 at McGill, Dr. Merle has earned numerous fellowships and awards, including support from the Dobson Foundation for Surgical Innovation and the prestigious FRQS Junior Research Scholar Award. So please join me in welcoming our Professor for the talk.
Thank you, Professor Merle, for that nice introduction. And thank you for the invitation. It's a great pleasure to come to discuss a bit my research in your program. So yes, indeed, like in 2017, I was an assistant professor in the Department of Surgery. And I was attached to the division of orthopedic surgery. So that's why today I'm going to talk on how we explore in my group a new frontier for bone healing, patient outcomes beyond autografts. So I have no conflict of interest to declare, and I'm going to talk about a bit. So I have actually, I did study medicine and biochemistry, and I've done my PhD in fuel cell. Okay. So I was working on nanomaterial, catalysis. I did a lot of things during my background and my research as a postdoc. I worked on solar panel, on fuel cells. So you're going to see how my expertise and that I've gained during these years, I actually implemented into my research right now. So I have a group which is split in two. The first group is working on diagnostic and the second group on therapeutic. I'm going quickly to give you a snapshot on the diagnostic bit, but I will focus the rest of my talk on the therapeutic aspect of my research. So I did work on electrochemical sensor. So I don't know the people who knows what's an electrochemical sensor. So I'm going to explain you a bit what it is and what I've been doing in which application. So I work on musculoskeletal infection, so on bacterial infection, but I work as well. So I had two PhD students that worked on cancer screening. So you know that there is like 10 million people are dying because of cancers. One of the reasons is because sometimes the diagnostic, we don't have access to early detection and intervention. So currently the diagnostic is imaging or tissue liquid biopsy. The problem with those techniques, they're actually quite expensive, they're time consuming, and they are not always assertive. So my group, we try to develop sensors that I can actually tackle all these problems. So they are cheaper, they're fast, they can give you an answer in like 10, 15 minutes. And hopefully they are a bit more assertive than the current technology. So quickly, I'm going to explain you what's an electrochemical sensor. So this is a very schematic or an electrochemical sensor. So basically, you analyze what you want to measure. In the case of cancer, it's going to be a biomarker. It can be a lot of things. It can be like DNA. It can be protein. It can be cells. It can be as well peroxide and other different markers like glucose and pH and so on. Here you have receptor, so that's what you're going to work on. It's what's going to be able to identify to specifically your biomarker. Here you have your transducer. In the case of electrochemical sensor, it's usually the electrode. And that electrode after that is going to recognize the biomarker, and you're going to be able to measure a signal. Okay, the signal is electrochemical. That means you're measuring a current, so a flow of electron. So there is different type of, okay. You can measure, for instance, an enzyme, okay. For instance, in the case of cancer, you can measure an enzyme, for instance, like matrix metalloproteinase. They're like enzymes that actually allow cancer cells to progress inside the tissue, and their expression are upregulated in cancer patients. So you could measure an enzyme, okay. That enzyme are specific to a substrate. So they're going, during that reaction, they're going to produce some product and electron. You're going to measure those electrodes. So that's your signal at the end. It can be as well ion in solution. So I'm not going to go into details on that one. It can be as well like zinc finger protein. So a protein who has actually a redox group, something that can oxidize and reduce. So you can measure that reaction of oxidation and reduction directly at your surface of your electrode. Or it can be like using an antibody, okay? You know that antibody, they are specific to their antigen or protein. So if you know the biomarker is a protein, you can have like immobilize on the surface of your electrode. You could immobilize, for example, an antibody, which is going to recognize that antigen and then after that measure a signal. Okay, so that's a quick reminder to understand the following. So we worked on different things in terms of cancer. We work on metal. We know that zinc is highly concentrated in tissue, in cancerous tissue. So we developed a new technique. But again, it's really cheap material. We decided to work on cheap material and on non-biological compounds. So not immuno sensor, no antibody, because we know that antibody, they have a very short shelf life. So after like even like 4 or 5 hours, they will lose your activity. So we worked on really this simple chemical structure that we immobilize on the surface of the electrode. And the structure is here, as you can see, it's called zincon. They are extremely specific to zinc. They're going to chelate. So they're going to attach specifically the ion on the surface on this structure. And then you can measure the attachment of the zinc inside on your electrode. And here you can see the resistance of your electrode that decrease. Another way we can do that, as I mentioned, you can use enzyme. You may have heard about carbonic anhydrase. This is the enzyme actually you have in your mouth that give that tingly state. That as well facilitate the dissolution of CO2 into water and then create this balance with the bicarbonates. You have this matrix metalloproteinase and zinc finger protein. In that case, we work on a different approach. Again, something very stable. We worked on a drug which is specific of a domain present on this matrix metalloproteinase. So that drug is going to actually to attach to that domain, specifically of the MMP. And when it's attached, you're going to create a resistance on top of your electrode. And then you can measure that and see actually the concentration of that enzyme in your media. The last approach we did, we work on a new type of biological compound immobilized on electrode. It's called aptamer. So I don't know if anybody's aware or know what's an aptamer. So an aptamer is a single-strand DNA. Okay, it's a lot of nucleic acid linked together, and they are extremely specific to a protein. Okay. So here you can see your single-strand DNA. We immobilize that single-strand DNA on the surface of an electrode. And what's happening here, you have 2 different redox molecules. It's called methylene blue. You must maybe have heard about it. So this methylene blue, what they do, they can actually oxidize and reduce. And they have a very specific, we call that electrochemical signature. So when they're going to be in contact or at the proximity, at the vicinity of the electrode, they can actually give electron or take electron. Okay, so here you have a single-strand DNA. You can see that it has a specific structure. Here it's like unfolded, so it's straight. And the problem is in that structure is that the redox molecule here, they are very far from the electron, okay? Very far. That means that this molecule, they are not able to transfer the electron. But when you have this protein here, mucin 1, which is actually the biomarker for breast cancer, so when you have this protein, it's going to attach to that single-strand DNA, to that aptamer, and when it attaches, the aptamer is going to change completely its conformation. And when it changes conformation, it's going to take a folded state, as you can see. It's going to fold on itself. And when it folds on itself, you can see that the redox probe here, the methylene blue, becomes very close to the electrode. And as you can imagine, when it come very close to the electrode, it can transfer his electron. It can reduce and oxidize. So you can measure the signal. So basically, when you measure the signal is going to be related to the concentration of mucin 1 in the electrode. And we show that we were able to measure a limit of detection. That means that the minimum concentration of mucin 1 present in the blood that we can measure with this system is 0.32 nanomole. Okay, so it's very low concentration of mucin 1. We did the same as well with VGF, and this has been actually published. So if you're interested a bit more about this kind of diagnostic that we developed in my group, you can actually look on this paper and you can contact me. We can discuss about it. But today talk, so that was only like a small shot. I want to talk about therapeutic, and I want to talk about bone growth with the use of osteoinductive nanomaterial and cold therapy. I told you, I'm an electrochemist and I like nanomaterial. So that's what we did in the group. I'm not going to go into details about that, but you know that in some case, in some population, aging or people who suffer from obesity or diabetes, they have problem to heal bone when you have a fracture. Okay, so the gold standard is autograft, but there are some issues associated with this technique. So people have been developing synthetic material. And among them, you have the calcium phosphate. They are based bioceramics. They have shown some very good outcomes on bone healing. The problem is with those structures, they have like a very fast resorption and they have a very poor mechanical strength. So the idea was like, oh, maybe we can actually mix some nanomaterial who are pretty strong with those ceramics. So among them, there is a lot of nanomaterial, okay? And there is even more paper on using graphene. Graphene is like a 2D sheets of carbon. They're measuring a few nanometer thick and they have a very wide lateral dimension, okay? And they are usually exploited because they have exceptional electrical, chemical, and electronic properties. What has been shown is that this graphene actually has a very high tensile strength when it's mixed with bioceramics. It promotes protein absorption, cell adhesion, and growth, and it helps the integrity of the ceramics. The problem with that, there are some concern about there's no barioresorption, and they have some cytotoxicity because we're still using carbon. So what we did is like, oh, what if we could use something else? So something similar at this 2D nanographene, but something which is actually bioresorbable. And that's one of my master student, Rachel Monk, that actually graduated. She worked on hematine, which is actually a 2D layer of iron oxide. And you know that iron oxide is biodegradable. You can find some trace of iron in your body, okay? So what we did, we took hematite. It's very cheap. It's like a bulk. It's like a piece of iron oxide. And we developed a system to increase to scale up the production of these two dimension of graphene oxide. So here you have some transmission electron microscopy. So imagine it's really, really small material. It's like 0.5 nanometer, so it's really tiny. And you can see, so this is like 1 or 2 layers of sheets of iron oxide. And here you have the solution. what we did, so we took that solution and she mixed it with some implants, so some bioceramics. So you see that, first of all, the compressive strength increased, the pore size as well decreased because you kind of fill the porosity with your nanomaterial. Here it's an X-ray diffractogram to show that when you added the hematine, you didn't change the crystallinity of your implants. And then after that, we actually seeded cell on it to see if actually we could see some improvement by adding this 2D layer of iron oxide. And indeed, we show that here you have the cross-section and the surface of the monetite itself, and here it's when it stopped with hematine. So here you have the cross-section and the surface, and here somehow you're showing some mineralization, and you can see some cell and also some extracellular matrix already formed into the cement. And you can see that actually the expression pattern, so we measure alkaline phosphatase, which is actually an early marker of osteoblast differentiation. It was actually improved, upregulated in presence of hematine. And RUNX2, what's measured, it's a gene encoding for osteoblast differentiation. So these two actually results show that the presence of hematine mixed with monetine is improve the differentiation of osteoblast. Osteoblast being the cell that produce bone, that form the bone. So we were pretty happy about that result. And in the meantime, we saw that paper coming out with in the MIT review. Sorry, graphene, porphyrin is the new wonder material that got everyone excited. And it got us also excited, okay, when we saw that, because this graphene-like nanosheet composed of boron atoms, they was only used for application such as electronic, as you can see, optoelectronic, magnetic. It has amazing application, but nobody used them for biomedical application. And in the meantime, we had a student who was working actually on boron accelerating, showing that boron actually facilitate bone growth. So in that study, we actually create a bone defect. We create a cortical window defect inside the diaphysis of a mouse. And then we injected boric acid every 2 or 3 days, I don't remember, every 48 hours until euthanasia. And what we saw is that actually the bone was highly accelerated. And on top of that, so I'm not going to go through that actually, but we saw that we have so not only more bone, but as well an enhanced angiogenesis. So I put the paper here if you're interested. So we had that study and we had that paper saying that porphyrin is even more exciting material than graphene. And you know that boron is actually, you can find boron in your body. So it's actually biodegradable. So given the impact of this compound or this boron on the osteogenic response that we saw on the previous study, and we knew as well that boron deficiency show an impaired growth both in impaired bone growth and repair, was like, oh, maybe we could actually produce that porphyrin, dope it with cement, and get even better results than this injection of boric acid. Okay. So that's what we did. And this is the work of Justin Mata, who actually should be graduating before the end of the year. So he actually spent a huge amount of time trying to exfoliate porphyrin from a boron nanoparticle. And that's actually was a pretty difficult job, because what you want is to have a very big lateral dimension with just 1 or 2 layers of porphyrin. And at the end, he did it by playing with solvent and ultrasound parameter, and he was able to mix so those 2 layers, those porphyrin nanosheet with, again, monetite implants. So once again, when you had the porphyrin, you have no change in the crystallinity of your material. Again, you have a huge, you have a very high, you have a better mechanical strength of your cement with not much difference in terms of porosity and pore size. So here you have the scanning electron microscopy image of your implant, so before and with and without monetite. So this is without and this is with the surface and the cross section. And here you can see some borophene, nanosheets inside your cement. Then we seed some pre-osteoblast cell onto the cement, and we check if it was biocompatible and if the cell were able to proliferate onto the cement and in within. And and this is what we saw. We saw a big difference between the with and without the presence of borophene. And then we checked as well the expression of some gene specific of the osteogenic differentiation and maturation. So first of all, what you have to see is that ALP, so it's alkaline phosphatase, which is actually an early marker of the differentiation of osteoblast, is actually significantly different for in presence with the borophene here compared to the monetite itself or even the untreated cell. That goes as well in conjunction with what we see with the RUNX2, which is actually the gene which encodes for the differentiation of the osteoblast. But what is even more important, I guess, as a message to take away, is that you can see SPARC here, which is actually the gene which encodes for osteonectin. What you see is that gene actually that code for the non-collagenous protein, which is actually a marker for mineralization, and here the osteocalcin, which encode for protein, a bone protein expressed in mature osteoblast. That means that these two genes actually related for mature osteoblast. You can see that they are highly elevated in the case with borophene, especially for osteocalcin, which is actually 5-fold higher at 21 days. So not only you differentiate the osteoblast, but you have mature osteoblast able to mineralize the matrix. So in the conclusion for that first chapter is that we were able, first of all, to scale up the process to produce this 2D nanosheet. And I say that because it was not that easy, okay? It's not that you just put the material in ultrasound and you hope for the best that you get some material. It's really difficult to get something without breaking up the nanosheet and get small pieces. And it's not working on electrocatalysis where you just need a little amount. Here, you need a huge amount of material because you need to do your experiments in triplicate and you need to dope them as much as you can in your cement. So that was the first thing, and I think they did a very good job by improving the technique, the synthesis technique for those 2 nanomaterial. Then we showed that actually by mixing them with the bioceramics, we're able to enhance the mechanical properties of those bioactive cements without changing the crystallinity. And that's important because I don't know if you're familiar with cement, but depending on the crystallinity, depending on the ceramic you will have different resorption. Then we show that doping the monetite with those 2 nanomaterial, and I don't make any distinction between the borophene or the hematine, but we both saw that you have an increased proliferation and increased expression of ALP differentiation in the early for both, but especially for borophene in the late osteoblastic marker. So the idea of using this borophene and hematine are pretty important because compared to carbon, we managed to develop this biodegradable synthetic implants. So those work, they were done by 2 master candidates. So, but there's still some work to do, okay? It's just an in vitro study. It will be nice to actually do some like future step to take on. So study the biodegradability. I just told you it's biodegradable, but I have no idea how long it takes for the material to degrade. So that's something that should be done. Checking as well the effect of these ions, because you're going to release some ions, some boron, some bore, and so on, to have to see the effect on, for instance, on different signaling pathway. For instance, the Wnt beta signaling pathway will be interesting to check. And then after that, checking maybe, would it be possible maybe just to use, to use those nanomaterial for to differentiate mesenchymal stem cells, into osteoblasts. That should be done. And of course, finally, doing the in vivo. So that's the first conclusion. So now I'm going to go quickly to the last topic I want to go is the bone growth with cold therapy, because I guess it's a kind of cornerstone on our research. That's the first project that I've been developing when I joined McGill in 2017. And that's the story with the surfer. So that project has been done with Dr. Daniel Castano. He was a resident in orthopedic surgery. And he got that crazy idea that actually by using cold, you could actually improve bone growth. And he had that idea because he saw that surfer here, when surfer immerse their head very often in the water, you're going to create that overgrowth here of bone in the ear. That's called the surfer here. So he saw that, oh, maybe that we could actually replicate that inside the legs. That's what he thought. But when we looked at all the report, actually, we saw that environmental cold actually had a horrible negative effect on bone growth. So we were very suspicious about it. So we create first this bone defect model, the same as the other time. So we create that cortical defect in the diaphysis of the leg of a mouse. And what we did is that we close it up, and then every day for 24 days, as you can see here, we took the mouse, and then we just soak her legs for a certain time in cold water. Okay. And what we saw, we're actually amazed, is that not only you can see that the bone was completely filled, much faster after 24 days than the control, which was not immersed in the cold water, but we have as well much more blood vessel. So that was good. But it was like, okay, it's just a bone defect, is not clinically relevant. That was the feedback we had all the time from clinicians. Yeah, it's a bone defect, is not a fracture model. So here comes Matthew at the beginning of his PhD, as you can see here. And he developed a fracture model, okay? So here you have the guillotine, and what he proved that it was not only the bone defect model, the cold actually enhanced bone growth, accelerate the bone growth as well in the fracture model, okay? So that was really strange because as you could read everything about cold, everything about like chronic cold showed that you have a delay in the bone growth. So if you check with the time as well, it looks like time matters. If you go above 15 minutes, you can see that again, the bone volume decrease. So that was the first things, like acute cold could actually stimulate the bone healing. And that's a picture of Matthew in the middle of his PhD. You can see that he got a bit crazy during his PhD. Okay, and you're going to see in the second part how it is. So I'm not going to show you like a million staining and some graph, but I'm going to explain you why is it complicated, why cold healing is a complex mechanism. And that's the last study that actually Matthew performed. So what we saw is that we have the bone volume increase, okay? We could see that on both, not only on the cortical defect, but as well on the fracture model, that the bone volume increase and the angiogenesis as well was stimulated, was activated. So first, if we check on the bone volume, what we saw is that upon cold exposure, but I mean very acute, is only 15 minutes exposure to cold, we saw that we have upregulation, an increase in the detection of those acute cold shock protein. And those acute cold shock protein here, they are responsible for the upregulation of the osteoblast differentiation. And we did see as well an increase in ILP, so the early marker of differentiation, but as well an increase of osteocalcin, which actually indicate the presence of a mature osteoblast capable of mineralization. So that was the first thing. But we saw as well angiogenesis was stimulated. And that was actually it was difficult for us to prove why, because we couldn't figure it out. So what we saw is that we have an elevated, so we measure the hypoxic cell upon cold exposure, and we saw that you have a drastic increase of hypoxia near the injury site upon cold. And that was associated with an increase on VEGF, but as well on a marker for endothelial cell, the CD34. And we saw that these two things were in conjunction with actually a modification of the vasomotor tone through TRP-related pathway, okay, which are actually a transient receptor protein channel. So there are still some things to do, okay? We still find we have other like avenue to look into. So there will be like elucidating the mechanism will be a good things to do. Identifying the optimal window. Right now, we just do like one immersion every day. Maybe it is not the best, maybe one, maybe you need to stop at 7 days. So there is still some like optimal, there is still an optimal window to identify. Translating these to maybe a large animal model, that would be great. And as well, designing a cold system delivery. You can imagine we're not going to be able to take someone's legs and to immerse in cold water. Although we are in Canada, we could do that. We need to design a system for that. So I hope I show you that cold effect has a multifaceted effect and that actually could offer some insight for novel therapeutic strategy. So even if I did not, I hope Matthew in the second part will be able to clarify that. So I would like to thank all the people that work on that project, especially the people in the lab. They are the ones that do all the work, all the funding agency and my collaborator, and you for your attention, sorry. And we'll be very happy to answer any of your questions.
Thank you very much, Professor Merle, for that wonderful presentation. Please feel free to write in the chat or ask, just unmute and ask.
We can take the question at the end because Matthew is related anyway, Stock.
Okay. Matthew, you want to go next and then we ask the questions at the end?
Sure. Yeah, I could do that.
Okay.
And you can start to write them on the chat if you want then.
Yeah.
Just a second. Okay, perfect. One moment. Perfect. Everybody can see my slides?
Yes.
All right, so pretty much my name is Matthew Zakaria, and I'm pretty much going to be going off where Geraldine Merle left off and pretty much go more into the cold therapy regime, especially what's called with PTH was an interesting discovery that we kind of looked into, especially when it came to fracture healing. So beforehand, I have nothing to disclose. And before I begin, I'd like to acknowledge a few people that were really crucial when it came to everything. I'd like to acknowledge my supervisors, Dr. Geraldine Merle and Dr. Edward Harvey, as well as my colleagues and collaborators that contributed towards this project. So kind of just to recap on what Dr. Merle went over, our preceding studies showed that there was a facilitation of the fracture repair process following daily cold therapy, in which there was enhanced mineralization, especially within the fracture callus region, 14 days after treatment, and earlier signs of bone remodeling up to 28 days after treatment within a Miriam model. This led us to consider possible mechanisms that may explain to the increase in mineralization and to further dig into how that might come to be. And that's where we looked into calcium sensing receptors and their presence within the bone injury site. So with collaborators at the Lady Davis Institute, our preliminary findings showed a strong increase actually of these calcium sensing receptors in the injury site, and they're pretty much essential when it comes to the uptake and deposition of calcium in new bone formation. So just to give a little bit of information on calcium sensing receptors and their importance to the human body. Calcium sensing receptors are essential in maintaining extracellular calcium in diverse tissues such as parathyroid glands, kidneys, and bone. It controls PTH secretion, which in turn, via direct and indirect actions on kidney, bone, and intestine, maintains a normal extracellular concentration. So specifically, these receptors in bone tissues modulate the activity of osteoblasts and osteoclasts to regulate bone metabolism. Recent studies have shown that the activation of calcium sensing receptors actually causes a proliferation and differentiation of osteoblasts to accelerate bone fracture repair. Now, PTH do not activate osteoblasts directly, but rather through indirect activation by initially activating osteoblasts through osteoblastogenesis through the release of rankle, which is important in the differentiation of osteoblasts. So for a brief period of time, a bone formation environment is formed. So this ability to create a greater anabolic window with bone-forming activities of osteoblasts to a greater extent than the bone-resorbing activities of osteoclasts is one of the primary therapeutic advantages of administration of PTH. PTH, as well as an FDA-approved drug, producing anabolism in osteoporotic skeletons and shows a potential as a bone-building agent on fractured bone. However, this therapy has not been exactly optimized, as the maximum allowed dosage is 20 micrograms per kilogram, as anything more leads to potential hypercalcemic and other side effects, including the formation of bone cancer. So to look into the entire aspect in a more pathway-based way, the incorporation of PTH has shown to initially induce osteoprogenitors that become osteoblasts, thereby responsible for chondrocyte formation, and osteoblast differentiation, which leads to bone formation and calcium uptake. However, PTH's native function is accomplished by, or once rankle is released from osteoprogenitors and early forms of osteoblasts that strongly activates osteoclastogenesis and the maturation of osteoclasts that leads to bone loss. Additionally, prolonged or high dosages of PTH leads to the release of endogenous PTH that thereby creates a thereby negates excessive bone resorption, creating the cycle of bone resorption and absorption that leads to a harmful hypercalcemic environment from the release of too much calcium into the blood. So we think that, well, at the time we thought that by incorporating pole therapy, we postulated that osteoblast differentiation will be enhanced and accelerated through calcium sensing receptors, which will have a prime role in increasing the uptake of calcium within mature osteoblasts to achieve better mineralization within the bone injury site and consequently mitigate the formation of a hypercalcic environment. So as a result, the hypothesis we went into this study with was that pole treatment can leverage the osteoanabolic properties of intermittent PTH by stimulating CASR, or calcium sensing receptors, thereby expediting the process of fracture repair. So the methodology we went by was the creation of a bilateral fracture model using that intimidating guillotine that we created. And this was done within all the mice, and all the treatments consisted of a regime that lasted for 14 days. Four groups were created, one with no treatments to serve as a baseline, one with daily PTH injections of 40 micrograms per kilogram, one with daily cold treatment on the experimental hindlimb, accompanied with daily PTH injections of 40 micrograms per kilogram, and lastly, one with daily cold treatment on the experimental hindlimb. The cold treatment duration specifically lasted for 15 minutes daily, and the body temperature was maintained with a heating pad. Blood was collected at 7 days and 14 days through saphenous vein collection to get blood from as close to the injection site, as close to the injury site as possible. Bone morphology was also looked into to look into the formation of mineralization within the fracture region following the treatments. So interestingly, we were able to show that cold exposure actually led to reduction of the concentration of calcium in blood within normal levels after PTH injection, indicating that an uptake of calcium following cold treatment, and that cold application safeguards against PTH-induced hypercalcemia, thereby creating that acceptable normal range that would thereby negate those side effects that we wouldn't want to see. Then when we looked into bone morphology, our results revealed that concurrent use of cold therapy along with PTH treatment enhances the skeletal anabolism, and that PTH only treated mice showed a larger soft callus, indicating that there was facilitation of the chondrogenesis process and the formation of that fibrinogenous callus that precedes the formation of a bony callus, resulting in non-mineralized new bone. So the incorporation of cold, however, led to a recovery from the mineralization of new bone from PTH-induced chondrogenesis, and thereby increasing the amount of mineralization present within the injury region. So we also wanted to look further into ALP and calcium sensing receptor staining within the region. And right now, preliminary data demonstrates that cold stimulates the activation of calcium sensing receptors, which coincides with increased presence of ALP, an important biomarker of differentiating early osteoblasts. Collectively, this kind of demonstrates that enhanced mineralization potential within the bone injury region, following the combined application of cold and PTH is a possible regime to consider when it comes to facilitating bone healing. So just to kind of give a little recap on the entire project, but the findings from the study pretty much show that cold therapy can be utilized to uptake excessive calcium released by stimulating osteoblasts, osteoblast differentiation responsible for enhanced mineralization potential, and thereby negating the hypercalcemic side effect of PTH. Additional studies also need to look further into the calcium sensing receptor function, as well as other possible ways that cold and calcium sensing receptor function are directly related, and to look more into that specific pathway to give more clarity on that. Because even with all the literature that we have within our own hands and that's available, there's still a lot of conflicting findings in it, and especially when it comes to finding or creating that line between what accounts for acute and chronic cold exposure, especially as you consider larger animal models and different other parameters to consider, like even skin or the depth of skin and all of that. So that's something that needs further assessment. And also pretty much like the entire combination of cold therapy with intermittent PTH allows for another consideration for an interesting regime to be considered when it comes to approaching fracture healing. So with all that, thanks for listening, and we'll be taking any questions.
Thank you, Matthew. Please feel free to ask any questions. So I just open the, Geraldine, I like the whole sensor technology, really wonderful.
Which one? Sorry?
Sensors.
Oh, okay, the sensor, sorry.
You explained at the beginning. So I like the way the integration with aptamers.
Yeah.
So when you integrate with aptamers, with the electrochemical sensor platforms, how do you stabilize the signal and reduce the noise?
That's actually a pretty good question. You sure you're not working on sensor, Faxon? Do you want to can share my slide again, or maybe I don't need to? But yeah, I think I need to. So I really went very fast on that because okay, you see my slide, yeah. So what I haven't seen is here. I show you that's what we did. So there was a PhD student Ashkan who worked on that. He did all his PhD on how to optimize the signal. So you want to have a very high signal. You want to measure as much as you can, and to avoid all the background. Okay. And there have been lots of strategy to do that. So increasing the number of, but the problem is that it doesn't work very well. So what he did, his strategy was like, instead of what usually people do, they only put one redox molecule. So it's going to have like, let's say, one electron transfer. So what he did, he added two. So we did some chemistry on top of this DNA, and we attached two molecules instead of one. Right. So to multiply the signal by two, let's say, and that actually the signal to noise ratio is going to be increased by two. So you have less background. So that's what actually his PhD was about, to show that you can actually attach two without damaging the structure of the aptamer. And that works with both cases. So I haven't mentioned that, but here in that case, we go from unfolded to a folded states. But some aptamer worked on the other way. So you start folded and then you unfold. So you have a gain loss. That's a gain. So, and he showed that in his PhD that he could do both of them. He could actually modify, and you can do all this folding and unfolding on both directions.
That's neat. Really nice.
Yeah, thank you. I think there's a question in the chat as well.
The question is, from Abikumaran. Do you want to ask it?
Yeah, I just need to stop the sharing because I don't see. Yeah. Thank you, Laura. What's the optimal duration, frequency, and temperature of cold exposure? I think I could answer, but I think Mathieu, do you want to answer? That's your PhD project.
Yeah, I could definitely take this one. So pretty much 1 second. Yeah, so when it came to the optimal duration, frequency, and temperature of cold exposure needed to effectively accelerate bone healing. So pretty much for that, especially what we need to consider is importantly the animal model. So in this case, we specifically used a mouse model. So we have to take that into consideration. And what we were able to show was that at 15 minutes specifically, we had a better what's called better healing rate in comparison to the 10-day and to the what's called to the 10-minute, the 5-minute, and the 30-minute time that we also did. And so pretty much, I guess to answer that question, it was at the 15-minute mark, and the temperature that we got internally, which we were able to measure at the bone site, was 19 degrees Celsius, and that was shown to be the ideal temperature for facilitating bone healing. So for your second part, the local cold plasma therapy question, would that be as effective as direct localized cold therapy in enhancing bone repair? So that's something that, in that case, you'd have to look into the temperature attained at the bone injury site, as well as the duration as well. And to my knowledge, there aren't any studies that look into that specifically within an in vivo model, but it's something that could be looked into. And I think the main way to go about that would be, as long as it's something that's acute, if it's an acute type of approach, it could potentially be a way to facilitate bone repair. But it's all about creating that barrier of what counts for acute and chronic.
Did we answer your question, Abikumaran? Yeah, it's okay.
Thank you very much. Thank you.
No worries.
What if
Yeah.
you used to assess bone growth and healing? Okay, should I answer, Mathieu? I guess it's again.
Okay, yeah, I could go for this. So some of the yeah, I guess it's just rapid fire here. But so yeah, when it came to specific ways we went about it, we specifically looked into into creating micro CT analysis. So we took X-ray images of the bone, and then from there we were able to kind of create a reconstruction model of the bone, specifically the injury site, and then from there we used one software, this software specifically CT analysis, and that allows us to go through specific bone parameters like trabecular separation, trabecular thickness, as well as bone volume, bone mineral density. From there, we were able to gauge just how well the healing was going on within the region. And just to give a little bit of a brief understanding of those parameters, so trabecular separation looks into the distance between mineralization within bones. Trabecular thickness looks into how thick the mineralization is within the injury, within the bone injury. And pretty much bone volume, pretty explanatory, pretty much listen to encapsulating the amount of mineralized bone that was formed within the bone injury. And another thing we also were able to do with this, the CT analysis aspect, was that we had one very smart guy, Dr. Jose Ramirez Garcia. He was a previous student, and he actually was able to create a pipeline, a specific script that if you run it on like the CT analysis custom processing, what it allows you to do is create a specific type of channel through which you can map vasculature within the bone by creating open pores and open channels and kind of reconstructing how the vasculature is within the bone injury region. And from there, we were able to kind of get a better idea when it came to vasculature development and other aspects involved. And lastly, of course, we did a lot of staining. The amount of staining that over 4 or 5 years now it gave us a good picture as to specific pathways that were involved, and more specifically into immunohistochemical staining as well, to look into specific biomarkers or specific things that were involved when it came to differentiation of particular pathways. And yeah, that's
I think
it involved a lot of things.
to answer, Matthew, I guess you forgot to check the quality of the new bone. So we did as well some biomechanical assessments. So you check the hardness of the bone and so on. So that's as well something you do. So you try to do all what is non-destructive at the beginning, like CT image and so on, to visualize. Some mechanical testing, because we have the machine which actually doesn't destroy the bone. And then after that, you embed your sample, section it, and then you stain it to visualize more like markers. Did we answer your question correctly? Do you want more explanation?
So, Geraldine, I know this is a tough one. They ask it a lot. When doing things like what you are doing with the borafin monotide. So, what is the expected degradation rate of borafin monotide implant, and how does this align with typical bone healing timelines? So does it do they?
Yes. So the issue right now was actually to get, how we did the implants, because we didn't have a lot of material, I say we'd have more than before than other people, but still it's not enough. So we cannot mix it.
Right.
We cannot mix it with the monetite and then that's it. So what we do, we kind of like use a centrifugation process to force the 2D nanosheet to go inside. The thing is that after 1 week in media, you can already see by the naked eyes that the hematine is completely gone from the implant.
Okay.
Doesn't mean that it's degraded, but it means that it's not there.
Okay.
So that's something we need to figure out how to do the biodegradability.
Okay.
Is try to see how we can stick it on it, or maybe, using, I don't know, dialysis bag.
Right.
But to keep it in the solution that we can measure it, because at the moment we cannot measure the degradation. It just detach. But you can already see that after, even if it's 7 days on the implant, you can already see that he has like some very important effect on the cells. So if you find a way how to keep it in, maybe putting a coating on it, or I don't know, maybe sinter it a bit, do like a cold sintering, just to attach it, maybe to make stronger attachment. That could be an alternative to do, yeah.
That's
Did I
fascinating.
answer your question?
Yeah, that's really fascinating. I think
if we could keep it like for more than 7 days, like 24 days at least on it, but I don't think the degradation is going to be like a week. I think it's going to be more like months, because there are still metals.
Right.
So I expect, to get like the surface oxidized slowly first and then to be released. So I guess like 9 months, 12 months.
Right.
Which is okay, I guess. Better to go slowly, to release like a slowly dose of those material in your body instead of everything at once.
I think there's another question by Fatih.
Sorry. Will the approach you're using for bone healing potentially be applied to other fields like cartilage regeneration? Which approach? The stuff we're doing in cold, or just want to be specific? All the approach we try, or just the cold, or just the nanomaterial?
The cold one.
The cold one.
The cold one.
I think regenerating cartilage is a very complex and difficult task. I'm not sure cold will be easily that translated. What do you think, Matthew?
So actually, there's been a few companies
Or
that
like nerve repair, for example.
say again, sorry.
Other field like a nerve repair. Just not the
Okay,
cartilage, but also the nerve repair.
To the extent of nerve repair, I'm not entirely sure. I wouldn't yeah, I wouldn't be able to answer that. But when it comes to other types of field as well, like more so ligaments or more so other aspects, not just bone, there's been some companies that already have prototypes that when it comes to the application of short-term full therapy to the facilitation of like ACL repair, MCL repair, and other different types of ligaments. So it's not restricted to just bone. And I mean, the entire concept itself, it kind of extends from how the application of cold has universally been known to lead to a reduction in the inflammation. So there's other different fields that take that aspect into consideration, especially when it comes to ligament repair. But as to the extent of nerve damage or a nerve repair, I wouldn't be able to answer that.
Thank you.
Thanks.
Thank
I do have a question, if it's okay to ask.
Not sure.
I'll start with you, Matthew, actually, if you don't mind. Yeah, sure. That was an interesting photo, by the way, in that Geraldine showed. Yes, the journey.
I wanted to take a picture of
Yeah.
him now.
There's a big transition there.
Yeah,
Yeah, a little bit more than cold therapy was going on, I guess, in that photo.
stop therapy.
Yeah, so it's a question related to the photo. So you showed one of your slides. You showed that there's an increase in osteoclast numbers, at least, in cold therapy and cold plus PTH. Just since the osteoclast, is involved in degradation of bone, right, resorption of bone. I was wondering, I mean, you wouldn't want that to go on indefinitely. Obviously, you want to stop that at some point. So is there an increase in the number of osteoblasts at that site, like that's aiding in bone repair to compensate for those increased osteoclast numbers?
So yeah, that's a good point. And I think one good thing to keep in mind is also the environment as well when it comes to when this is being administered. In a bone healing environment, ideally you would want that initial upregulation, formation, and differentiation of osteoblasts to help facilitate the bone mineralization process. And osteoclasts as well have their role too, especially when it comes to bone remodeling. And that's one thing that we were able to show actually when it came to the 28-day mark, there was an increase in trap staining, which thereby showed that there was that anabolic and that catabolic for the time window as to when it comes to anabolic formation of bone. So the thing is, eventually, like you mentioned, the cycle does catch up, but it's more so about optimizing the injury at the right time with the right conditions to kind of create the ideal healing environment. And eventually, as the effect wanes off, that's when we kind of go back to normal levels and are able to see kind of a normal range when it comes to osteoblast and osteoclast presence within the injury site.
Perfect, thanks. I guess for both Geraldine and you, Matthew, you should, I guess, Geraldine, you showed in your slide that there was an increase in hypoxia in the bone following cold therapy. So I was just wondering if you had any idea on how cold could induce an increase in hypoxia at the injury site or the fracture site.
I could answer that. So yeah, within the injury side, one thing it kind of ties back into how we looked more into transient receptors, the TRPs, where that's TRPA1 and TRPM1. And both of them are very essential when it comes to vasoconstriction. And within the bone injury site, you're able to show that there was an increase of those receptors, which actually ties into how that leads to a cascade that then creates vasoconstriction. And then that by itself creates less availability of oxygen at the site and thereby a hypoxic environment. And one thing that's also tricky when it comes to translating in vitro to in vivo, which is why we really wanted to create an in vivo analysis of this, was that in vitro, it's hard to kind of mimic the same conditions, especially within a bone environment. As say, for example, some conditions will use 20-30% in vitro, but in a bone injury site, the normal range for oxygen levels is about 3 to 4 to 5% within that range, depending where the animal and everything, of course. So yeah, we kind of look back into focusing on TRP receptors and to kind of clarifying and using that as a way to explain the presence and prevalence of hypoxia development.
Perfect. Thank you.
I think, Michael, there is something still to be done because we had a
Yeah.
very biased approach, as you know.
We did.
Yeah.
So there's still things to be done, either using like blocker or checking actually that the blood flow is decreased. And this is, don't forget, it is done on the bone defects. It's not done on the bone fracture, where actually the vasculature might be compromised. So that might be even different for the fracture model.
True.
So there is a lot of things that needs to be done still, like a lot. So Mathieu is finishing his PhD soon, so we might need a student quite soon to take over a very good student to take over that cold study, not advertising position.
Sure. I had a question actually on the borofine. So that is very interesting. Yeah, you presented very interesting results there. So just two questions. Mainly is one,
I can give you some.
yeah. So I was wondering, the bone that was generated after you added the borofine, so in the repair, is that similar in quality to normal bone, so uninjured bone?
Okay, so the in vivo study has been done only with boric acid,
Okay.
neutralized boric acid. So we just
Okay.
taking bore. We only did in vitro, so we have to do the in vivo study. So Justin is finishing, so we like going to publish that things, but we need to do the in vivo work. So
No,
we
that's
have
good.
no
That's
idea how
really
it will
good.
become, but it's really amazing, the results,
Yeah, it
especially
is.
for the time being on the cement.
And also, like, do you find, is there a limitation as to whether you could use this in the clinic in terms of upscaling the amount of borofine for application?
Yeah, that's going to be a problem.
Yeah.
Definitely, because you get like, but I mean, we get quite a lot at the end. He really, he had to really work on, because you have to work on parameters like frequency, like duty cycle. Like you don't want the stuff to overheat, to oxidize, to change the properties of the material. So I don't show it because it's a lot of material engineering there, but he did work on that. But we got some good yield at the end. And I think after that, you need to give the process, and you have process engineer to work on that and to make it even.
Yeah, certainly. Yeah. The last one is actually on the aptamer. So Faxon brought up some questions. That's cool. I had one related more to the actual nucleotide, a single, right?
Yeah.
The single strand nucleotide. So if we're talking about developing a sensor, these nucleotides, are they ones that you came up with, or are they ones that have been validated in the literature or by some company or some?
They have been validated by the literature. So we work on auto biomarker, so this VAGF and this mucin one. There are not that many right now because it takes a lot of time for them to figure it out which aptamer is going. It's done synthetically, so you basically produce like population of those single strand nucleotide, put them in the protein, and you check which one actually and specifically attached to the protein. So the research is just quite recent. There is not many work on that kind of things, especially on this. So there are not many aptamers used still.
Okay.
So it goes along with in research with biomarkers. So first they need to find which biomarker, and then to find which aptamer, and then it goes.
Yeah, because I guess the issue would be when you're developing that, how would you get away with any proprietary issues dealing with the aptamer itself if it's already developed by somebody else? Yeah.
We were more working on strategy, like, exactly like the question of Faxon, it was really how to improve the signal of those.
Sure.
You don't want to work with antibody because although they're easy, you can buy them and so on. They are not stable, they're very expensive. It's done synthetically and is really stable. So
yeah, perfect. Well, thank you.
Thank you.
Yeah, thank you very much, Geraldine and Matthew, for that very exciting presentation. I'd like to
Thank you.
The students so asking questions. Abikumar, Fatima, thank you very much, and of course, Dr. Grant. Can you So thank you very much for attending this exciting meeting. And
thank you. And I have my email if you have question or if you want to collaborate or anything, just can contact me. Always pleased to answer your email.
Yeah. Thank you.
Thank you very much, Professor Moale, for your invitation.
and bye.
Bye.
Bye. Thank you, everybody.